When we started work on cannabis about 50 years ago, there was essentially no interest. Nobody was interested in... Okay. Nobody was interested in supporting our work. We applied to Israeli, we applied to American sources, and we were told that cannabis is not relevant. Cannabis is used a little bit in Mexico, in South America, not in the US, not in Israel, so find something more relevant. Well, almost a year later, actually, the pharmacologist, the National Institute of Health in the US came over. Turned out that, yes, it had become relevant. People were using it, and NIH had no research projects, so they were interested in going ahead. Well, if we look today what's going on, there is a huge interest. As a matter of fact, Nature, one of the leading science journals, had 4 reviews on cannabis. You can see on the picture here, cannabis growing out of the brain, and there have been thousands of papers on cannabis. And the reason for that is, well, not very simple, but true. 2 investigators from NIH wrote a review, and they say that modulating the cannabinoid activity may have therapeutic potential in almost all diseases affecting humans, and they give a lot of references and a lot of information. Now, this is a very, very strong statement. I'm not aware of any other biological system that one can say that it is involved and there is therapeutic potential in almost all diseases affecting humans. So this is the reason why scientists in essentially every segment of biology are interested on the activity of the endogenous cannabinoids, this is why many of you are here, I assume. But let's go back a few years, 3,000-4,000 years ago. Actually, cannabis was used in China, in India, in the Middle East. This is something that the Assyrians used. They called it ganzi gunu, which is the recreational cannabis, the drug that takes away the mind. That's what they called it, ganzi gunu. They used another word for the medicinal properties, and they actually used cannabis in their religious rites. And it went on like that for a couple of thousand years. And I will not go into details of the history. I'll try to more or less present an overview of what's going on. And I can see, from my point of view, 3 phases of cannabinoid research. One of them has to do with the plant and the plant cannabinoids. And this is the phytocannabinoid research. Most people, both in science and in industry, are interested in the phytocannabinoid research, and THC and cannabidiol and so on. But there are other phases, and the second one, and I'll speak about all 3 of them, is the endogenous cannabinoid research, the compounds, the cannabinoid compounds that we make in our body. And there is a third phase that has just started, or very few researchers are involved in it at the moment. These are compounds which are closely related chemically to the compounds that we already make, cannabinoid, that they're involved in many other things, essentially, and probably most physiological reactions. So I will try in the few minutes I have to say a few words about all 3 phases of research. Let's start with the phytochemical work. Well, you'll see a lot of structures, but that doesn't matter. I'll try to present it without relating directly to the structures. When we started work in the early 60s, there had been work presented by researchers both in the UK and the US, but probably due to the paucity of techniques, the actual active compound had never been isolated in pure form, and its structure was known in a very general way, but not exact. Well, we found that there are a large number of compounds in cannabis. Today we know that there are about 100, and we isolated the major ones. Some of them were new, some of them had been isolated, but the structures were unknown. And here are a few examples, cannabigerol and cannabicyclol, cannabichromin, etc. But the two most important ones are cannabidiol and THC, tetrahydrocannabinol. We showed the structure of cannabidiol way back in 1963, quite a few years ago, and we isolated, I believe for the first time in pure form, THC and elucidated its structure. These two compounds, and I shall speak about both of them, have completely different biological activity, and they are actually not natural products. THC and cannabidiol are not made by the plant. The plant makes their precursors, the acids, THC acid and cannabidiolic acid, and we showed that again many, many years ago. Now, the problem with these acids is that they are not stable, and scientists do not like to work with unstable products because it is very difficult to reproduce work. So until quite recently, there was very little work on the actual products that the plant makes, the THC acid and the cannabidiolic acid. So are these compounds active? The answer is yes. We just published, you can see two, that was of the last few months. We were able to stabilize these unstable acids that are present in the plant, and we found that these acids are very potent. They may be more potent in the case of cannabidiolic acid being more potent than cannabidiol. About THC, we're not sure yet. We're working on that. But cannabidiolic acid was found to suppress nausea and suppress anxiety. And cannabidiolic acid also lowers depression, at least in animals. So here we have a new field of research, the acids. And as I said, very few papers, and chances are that they will be more widely used than their breakdown products, because they are at least as active and probably more active than them. Now I would like to say another word about the two compounds. In the plant, of course, you know that there are a lot of other compounds available, not cannabinoids necessarily, and these compounds affect the activity of THC, possibly of cannabidiol. We call that effect the entourage effect, and it has not been investigated thoroughly. The term is known and people use it, but there has been very little work on the entourage effect, and not all effects of THC or cannabidiol can be changed by the entourage effect. So again, here is a wide field of investigation, and I hope that we shall know more about the entourage effect over the next few years. Now, what are the physiological systems and conditions affected by cannabinoids, the plant cannabinoids? And here I have a partial list, just starting from anxiety and going down by ABC, blood pressure, digestive system, etc., mood, movement, down to stress. So we have to know more about each specific one. Is only THC involved? Are both compounds involved? Is only CBD involved? Is the entourage? So we have to learn quite a lot about these things. Now, what are the medical actions of THC? I will not go into detail. This is quite well known. It is being widely used for pain, certain types of pain, not all kinds of pain, not acute pain, for example, but many of the other pains, yes, most definitely, and it doesn't cause the side effects of opiates. Of course, it has been THC approved, both in the US and here, for the side effects of cancer chemotherapy, nausea and vomiting, and there is work by our group, by other groups on post-trauma. THC most definitely affects post-trauma. So here we have a drug that is used not widely because of the side effects. But the more important constituent, of course, as we well know, is cannabidiol, CBD. Many, many years ago, you can see the date, 1980, we did, after work on mice that cannabidiol is of importance in epilepsy, and it was a double-blind study which we did in collaborators in South America. And we found that, yes, most definitely, when cannabidiol in pretty high doses, 200 milligrams more or less per patient, were given to patients, epileptic patients, that nothing else was helping them, yes, cannabidiol helped tremendously. 4 of these patients had no attacks, 3 of them had very few attacks, and only 1 was not helped. So here we had something published in 1980 showing that cannabidiol is a novel anti-epileptic drug. We thought that it will be accepted by the neurologists and they will try to go ahead, but then nothing happened for about 30 years. We could have helped a lot of people, a lot of children. Unfortunately, nobody was interested, nothing happened. Then about 10 years ago, parents found out that, that cannabis with a lot of cannabidiol, and maybe our work helped understand that, helped children. They started in the US going down to Colorado because cannabidiol was available in plants there. And yes, it was found that plants with a lot of cannabidiol helps children with epilepsy. So there was a public pressure on the appropriate US agencies. Large clinical trial was approved, and it showed, yes, cannabidiol in about 60% of patients in certain epileptic diseases. Epilepsy is not one disease, epilepsy is several types of diseases, is extremely helpful. And cannabidiol now, under a different name, has been approved for epilepsy in children. Again, it could have been done 30 years ago. Well, it wasn't. And today, there's a strange situation. Cannabidiol is still not approved, in the US at least, and not here, and it is next to heroin in the non-approved drugs, but under a different name, it is approved as a drug. I mean, I'm not a legal person. I'm not sure I understand how the same compound can be both on the top, very dangerous, and down, yes, it can be used as a drug. But this is where we have it at the moment. Now, in Israel, a group, I was a little bit involved with that group, looked at graft versus host disease. This is a complication that occurs after bone marrow transplant. Bone marrow transplant has to be done, certain cancer patients, and then the body attacks the new bone marrow, the bone marrow attacks the body, and the patient is sick, very sick. Dr. Yeshurun found that administering CBD, cannabidiol, large doses to patients, it was extremely helpful, and today it is being developed by a company. I hope that cannabidiol will be used in host versus graft versus host disease. Another field, again with a lot of question marks, is schizophrenia. A German group found that cannabidiol in a double-blind study was very effective. It was compared to a potent antipsychotic agent, amisulpride, and both drugs were very effective, with amisulpride causing side effects and cannabidiol causing no side effects. Again, I thought that it will be used, accepted. Well, it is not. No, it is not accepted neither this country nor in any other country that I'm aware of. I have no idea why not. Schizophrenia is a disease that attacks about 100% of the population, and it is a major, major problem in essentially all the countries. Why cannabidiol has not been advanced, I have no idea. Another disease is diabetes type 1. Cannabidiol is a very efficient agent in autoimmune diseases, diseases in which the body attacks itself for a number of reasons. We don't always know them. So one of the diseases is diabetes type 1. And in diabetes type 1, we have shown that it is a very effective agent, cannabidiol. And yet, although the mechanism is exactly the same, both in mice, at least in the model we had, and in humans, it is not being developed in that field. Why not? I have no idea. Well, I want to mention now derivatives of cannabidiol. Many of the natural drugs we have are not used as the natural drugs themselves, but are used as derivatives. You cannot go to a pharmacy and get penicillin. You can get derivatives of penicillin. You cannot get cortisone, one of the human hormones. You can get derivatives of cortisone because they are active at a lower better in many other ways. So one of the things we did some years ago, and this compound, the new compound, which is made as a fluorination of CBD, and that is fluoro CBD. It was licensed to a company, Phytochemical. This compound is more active than CBD, so chances are that in a few years we shall see derivatives, maybe including this derivative, in derivatives of cannabidiol, which may be potent or more potent than cannabidiol itself. Now, here I spoke about the phytochemicals, the compounds found in the plant. For nearly 20 years after the discovery of most of these compounds, after the discovery in the 60s of THC and so on, there was a lot of work on physiology, pharmacology, chemistry. One thing was not known, and it was, what is the mechanism? Then a group in the US found that there is a receptor, a protein that THC binds to, and this is the way it acts. Now, we don't have receptors in our body because there is a plant out there. We have receptors in our body because our body makes compounds which activate, stimulate, or block a particular receptor. So we went ahead looking for such compounds. And we thought that, yes, the receptors are relevant because they are found exactly in those regions of the brain that are involved in cannabinoid effects, like regions in the brain that have to do with movement control, with body movement coordination, with learning and memory, etc., etc. So we worked for a few years, and I will, of course, not go into any technical details, and we were able, after a few years of work, to identify a compound, and this compound was exactly equivalent to THC in its actions, except that it acted and its action disappeared because it was broken down. But otherwise, it was essentially the same as THC. We called it an Endomite. Their structures were completely different. THC is one type of compound, and those of you who remember your chemistry will see the one on top, the THC. And an Endomite, on the left-hand side, is completely different chemically, but they have the same type of activity, and both of them are liposoluble. They do not dissolve in water, they dissolve only in lipids. So, in this respect, they are equivalent. And a few years later, we found a second compound, which we call 2-AG. So, our body makes an Endomite and 2-AG, and these compounds are the compounds that act on the receptors that THC acts upon, and they're equivalent to them. And there is a lot of work that has been done on these compounds. As a matter of fact, our paper on an Endomite has been cited thousands of times because it's an important neurotransmitter, a new novel neurotransmitter. Now, I will not go into the biochemistry, except one thing. Our nerves are not just going, let's say, from my foot to the brain. They're always broken down by what we call the synapses, and the synapses exist in order to make possible for the body to regulate the activity of nerves. And there are presynapse, there is a postsynapse. Now, the cannabinoids are formed when and where needed in the postsynapse, the second part of the nerve. When they are formed, they go to the presynapse, they act on a receptor, and then they act on other neurotransmitters. So, and the endogenous cannabinoids have activities of their own, but they also have activities by modulating the activity of other neurotransmitters. And this is why they're so important, because they can activate a large number of other neurotransmitters. So here we have a situation which explains why endogenous cannabinoids are so important. Now, the endogenous cannabinoid system involves these two compounds which we've had, an Endomite and 2-AG. It involves the receptors. We have two of them, CB1 and CB2. The CB1 receptor causes the effects of marijuana that we know, changes in mood, and changes in activity, and so on. The CB2 receptor is essentially a receptor that parallels the immune system. It helps, in most cases, helps the body defend itself in various ways, and it is probably of a huge, huge importance, and therefore quite a few companies are working on compounds that only act on the CB2 receptor, namely the receptor that helps. It is the immune-type receptor, and Hoffmann-La Roche in Switzerland is just publishing a paper on a compound that acts only on the CB2 receptor. I believe that many other companies will be going down the same way. Now, our body has to regulate these two endogenous compounds, so it has enzymes that break these compounds and enzymes that form these compounds. And again, there is quite a lot of work on these compounds because through them we can regulate the endogenous cannabinoids. So chances are, again, that within the next few years we'll see quite a lot on the enzymes that break the compounds and enzymes that form the compound, and by playing with them, we shall be able to regulate our endogenous cannabinoid system. Now, what does the system do? As I mentioned, quite a lot of things, and one of them is, let's say, neuroprotection. If there is brain damage, we see that the 2-AG goes up tremendously. Well, does it go up because the brain has been damaged, or does it go up because the brain tries to help itself? So we synthesized a lot of 2-AG and gave it to a poor mouse that had a brain damage, and we saw that, yes, it most definitely lowers the brain damage. You can see that on the left-hand side, pictures of the brain. In the control, there is twice as much damage than in those parts of the brain of a mouse that was given 2-AG after the damage. So 2-AG and the other endocannabinoids may be helping with brain damage, and indeed, only in the last few weeks, we are contacted by football players, the National Football League, the US, saying, can we do something because some of our players have concussions? Well, maybe. These are some of the diseases that CB2 receptor helps, and heart problems, atherosclerosis, stroke, liver, neurodegenerative diseases, and pain. So it is, as I said, an important part, and this is the second phase of cannabinoid research, the research on the endogenous compounds. So I'm going now to say a few words on the third part. Our body makes about 100 compounds that are very closely related to the endogenous compounds I was speaking about. Now, why does the body make them? Well, let's start a little bit, go back a little bit. We found that on brain injury, the endogenous compounds are very important. But they are not as important as we thought they will be, and we found that actually total extracts, the brain may be more helpful. So we thought maybe there are additional compounds there, and maybe they act on the vasoconstriction. When there is brain damage, there is a vasoconstriction, and maybe they help with the vasoconstriction. And yes, we found, after quite a lot of work, that there is a vasoconstriction. Now, we thought that these compounds should be the same as the endogenous cannabinoids. Our brain, our body is lazy. When it learns to make something, it will do it, make a small change and do something else. So it serves itself energy. This is well known. There are steroids, and this is a steroid. Forget about the structures and so on. By making small changes in the steroid, we find a lot of new compounds. So the system, our system is lazy. It tries not to do work that it doesn't have to do. It makes a compound, and then by making small changes, it makes a lot of additional compounds, like in the steroid. So here we had the same way. Endomite was made in the brain as an endogenous cannabinoid. Our brain, our body makes additional compounds, and in the case of vasoconstriction, it makes arachidonyl serine, a very closely related compound chemically. And we saw that in quite a few other systems. I'll give two examples. One of them is bones. It was well known, is well known, that women in the Mediterranean region have less osteoporosis than women in the north. And it was thought that this is due to the olive oil that is being used in the Mediterranean. So we looked at the olive oil itself. There is something called oleic acid there. It didn't help, but we found, after quite a lot of work, that actually a metabolite of olive oil is formed, and it is again closely related to endogenous cannabinoids. Again, the body doesn't like to work too much, so it takes something that it knows how to do, makes a small change, and we found that our body makes an anandamide compound, which we discovered the structure. It's called oleal serine, and this compound helps in bone formation. And as women in the Mediterranean region take olive oil as most of their oil, they make oleal serine, and this oleal serine helps with bone formation. And indeed, if we took a mouse and we caused loss of bones after by some medical ways, and the red line that you can see is the bone going down after being so treated in a mouse. If we give the oleal serine, the bones start forming again, and you can see the red one going up. So here we have, again, another example of how the body plays around with the endocannabinoid system, going into various directions. And now I finish with something that we have been doing over the last few months in collaboration. In these research projects, collaboration is essential. No lab can do everything involved with these effects we are looking at. One needs a chemical lab, a biochemical lab, a pharmacological lab, labs that have different interests and different experience. So in the field of addiction, just as example, there are 4 labs involved in addition to mine, a lab in the US, a lab in Canada, and a lab in Italy. And we thought that addiction is a disease. And if our body understand that addiction is a disease, then chances are that it will find ways or try to find ways to attack addiction like any other disease. It's not always successful, but it tries to do it. How does the body do that? Does it make compounds? We thought yes, and we looked at parts of the brain that are changed, or the synthesis of compounds that are changed and addiction, and we found a compound, and we found a structure, and this is again structure closely related to the endocannabinoids. It's oleal glycine, and it lowers very significantly the addiction to nicotine, and it blocks to a large extent the withdrawal symptoms both to nicotine and to opiates. If we take a mouse that has been addicted to nicotine and then gets no nicotine, it will not have the withdrawal effect that we always see. The same is true for heroin. If we take mice that are addicted to heroin, and we give oral glycine, they will not have the withdrawal effects that we normally see. Is that true for humans? We haven't got the foggiest idea. I think that it is true. If this is indeed true, then the company that has taken over the license on the compound and is developing it will have a large market to prevent withdrawal symptoms. Well, this oral glycine has been found to block nicotine withdrawal and nicotine reward, and this is a paper we have in press. Oral glycine has been found to affect morphine withdrawal. And to summarize my talk, we have the endocannabinoids that are involved in a large number of physiological processes, and THC mimics their action. The cannabidiol derivatives, which act by many mechanisms, may lead to a wide spectrum of novel drugs by itself. Cannabidiol is a major drug, or derivatives. Endocannabinoids can help us understand a lot of biological processes which we do not understand at the moment. And the fourth thing is that the CB2 specific agonists may be novel drugs in a wide range of diseases. And I want to thank my collaborators in Israel. You can see and those in Jerusalem that were in my lab, and colleagues in Jerusalem with which I worked and at other places, and a lot of collaboration abroad with people in the UK and people in Germany, in Switzerland, so on and so forth. And we collaborate closely, we visit each other, we drink a lot of coffee together, and I hope that we shall continue doing so for the next few years. Thank you.